Hi, welcome back to chapter 22, part 3. I just really quickly added this part right here, so I did not end my last lecture showing you this slide. So sorry if that one ended a little bit abruptly, but we are going to now talk about what happens when you don't have geographic isolation, but you still have speciation. You might remember this slide from before where I talked about the ways that speciation could happen, either allopatric or sympatric. And sympatric means same country speciation, sim meaning same. So in this case, we're going to really focus on the situation where you don't have a geographic barrier, but you still have speciation and how that might occur. There's basically three ways that this happens. One is polyploidy, and polyploidy is very challenging to understand, so we'll spend quite a bit of time talking about polyploidy. Then we'll focus a little bit on habitat differentiation and with sexual selection. So let's go ahead and talk about polyploidy first. Poly means many, right? And ploidy, that's about chromosome number. So one way that you can have sympatric speciation is by having a mutation where the chromosome number changes. Basically, polyploidy is the presence of extra sets, not just individual chromosomes, but extra sets of chromosomes due to accidents during cell division. So we're going to be talking about cell division here. We're going to be talking about mutation, and of course that gets complicated. So if you are having trouble remembering how meiosis works, I strongly recommend you go back and visit that chapter where we spent a lot of time talking about meiosis. And we did talk a little bit in that chapter about what happens if you have a failure of cell division, of cytokinesis. So just help yourself out by going back and reviewing that if you're a little confused by this subject. Okay, so polyploidy again happens when you have extra sets of chromosomes due to accidents during meiosis. Can happen during mitosis too, but you don't pass your genes on to a next generation in mitosis, so we're going to be focusing on meiosis. Polyploid speciation is actually very common in plants, but is much less common in animals. And you're going to find out why that's true in this section of the lecture, but let me just give you a heads up that basically plants have this ability to have both male gametes and female gametes, right? Sperm and eggs. And because of that, because that's true, plants can figure out how to do polyploidy, whereas animals have a much more challenging time of getting around this very large mutational error. Okay, so let's start with the basics here, and that is, what does the cell division error look like? So remember what a diploid cell is. A diploid cell has two homologous chromosomes for every chromosome, right? We call this a pair, or maybe we call this chromosome number 1, right? And maybe this is called chromosome number 2, and this is called chromosome number 3. And since there are a pair of each of those, we have a total of 6 chromosomes. 2n equals 6. If we go through making sister chromatids, right, and going through the process of cell division, PMAT, remember prophase, metaphase, anaphase, telophase, but we don't have cytokinesis. What happens is that you are separating your chromosomes from each other, especially your sister chromatids, but you're not forming a new cell. And also, just a quick reminder how important I said it was that cytokinesis is a separate phase from meiosis itself. So we count cytokinesis as a separate phase. And this is part of the reason why. So we have chromosome 1, and we have chromosome 2, and we have chromosome 3 here, but instead of having 2 members or a pair, we have 4 members. And so we say this cell is tetraploid instead of diploid. di meaning 2, tetra meaning 4. So instead of having 6 chromosomes, we have 4n equals 12, right? And that is a lot of chromosomes to have. Now, if this tetraploid cell, and remember its major problem was it didn't go through cytokinesis, right? If this tetraploid cell itself goes through meiosis, then what happens is that you have a gamete that is 2n, and these are the diploid gametes of a tetraploid cell. Now remember, plants can make both pollen or sperm and eggs, ova, and so these two could potentially fuse with each other. And if it did that, you'd have a new baby, a new individual, and this new individual is a whole new species. It is a tetraploid species. And this is, again, very common in plants. Actually, if you go to the grocery store, most of the plants that you see in the grocery store are polyploid. A great example of this is strawberries. And nice big juicy plump strawberries, right? And if you've ever seen strawberries that grow wild in your yard, you see these small, tiny little strawberries. And the main difference between those ones that grow wild in your yard versus the ones that people garden or grow on farms is that the ones on farms are tetraploid or polyploid of some sort, and the ones in the yard are not. So one of the interesting side effects of polyploidy in plants is that the fruits are much bigger than they are of the non-mutated type. So I've just stepped you through a very specific mutation and of polyploidy called autopolyploidy. And yes, you do need to know this term. Auto means self. So with autopolyploidy, you have an individual with more than 2 chromosome sets derived from one single species. So remember, we just had this one, and we had this error, and then we had meiosis, right? This is just one species. Yes, in the end, we have this new species that's tetraploid. It has 4 sets of chromosomes, but it happened within this one species. And it turns out that you can have polyploidy happen between 2 different species, each of which has a different set of chromosomes. So we use the term allopolyploidy. Remember, allo, we just learned this term in this chapter. Allo means different, right? Right, we talked about allopatric speciation. Allopolyploidy happens when a new species is formed from the combination of 2 or more different species over time. Allopolyploids can successfully mate with each other, but they cannot breed back with either of their parental species. Let's take a look at what that looks like. Okay, so in order to understand allopolyploidy, we're going to have 2 different species. So here's species A, and here's species B. And so notice that the diploid number for these 2 species is different. Our 2n is 6 for species A, and our 2n is 4 for species B. Now, if species A goes through a normal process of meiosis, it's going to make haploid gametes that have an n of 3. And for species B, that's going to go through normal meiosis, it's going to have a haploid number of 2. So far, so good. But, and here's the weird part, where you form a hybrid. The sperm and egg from A and B fuse together. Notice, and this is so important, that this hybrid, number 1, has a haploid number of 5. Why isn't it diploid? What's going on here? It's haploid because these don't have homologous chromosomes. And where's the homologous chromosome to this green one? it doesn't have a homologue with the purple one. And because it doesn't have a homologue with the purple one, we can't say that it's got a diploid number, right? It's only diploid if the homologue's present. So we say it's got a haploid number of 5. Because this individual cell right here is haploid, we also say that it's sterile. And it means that it can't itself reproduce unless something additional happens. And with Aloe polyploidy, that something additional that happens is a mutation. Now, we've already talked about this. We talked about this in auto polyploidy. Remember there was a mutation that, no, it wasn't a mutation. It was just a happenstance where cell division happened, but cytokinesis didn't happen. And that can happen for lots of reasons. But we're going to have the same thing happen in order for a new species to evolve. So in this case, the sterile hybrid is going to have a mitotic or meiotic error where it goes through prophase, metaphase, anaphase, telophase, right? But no cytokinesis. And if that happens, then each one of these makes its own copy. It makes its own homologue. In this case, the homologues are exactly the same because they used to be sister chromatids. They're just now unattached from each other. And so now this cell has a 2n number of 10, and because it's got its homologue there, it can go through normal cell division, and it can be, basically a whole new species. You might ask yourself how common this is. And the answer is, this is pretty common in plants. One of the things that we know about treating cancer, and again, just kind of letting you know how common this is, with cancer treatments, one of the side effects of some of the drugs that are derived from plants is that it kind of interferes with the cell division process. And if you don't remember this, go back to that chapter that we talked about in mitosis and review that. But if plant compounds can stop our cells from dividing properly, they can also prevent their own cells from dividing properly. And this does happen much more regularly than you might think. So Aloe polyploidy and auto polyploidy, these are fairly common in plants. Let's take a look at an actual example of this happening in nature. So we know of at least 5 new plant species that have evolved by polyploidy in nature since 1850. And the example that we're going to step through is the example of goat's beard, which has the scientific name of Tragopagon. Tragopagon. Say that 3 times fast. So here's what we know. These 3 diploid species have interbred with each other to produce 2 new tetraploid species. And this happened through Aloe polyploidy, but just like with everything biological, it's a little complicated because these are very, very closely related species. They all have the same chromosome number. So each of the parental types, each of the original species, have 12 chromosomes. And there they are right there with the gray. Their names are kind of in gray. So 12 chromosomes. And when this species, T. dubius, mated with T. perifolius, we came up with a 24 chromosome species called hybrid species called T. mirus. And again, the T stands for Tragobacon over here. The same thing, T. dubius mating in this case with T. pratensis led to the hybrid species of T. miscellus, and that also has a chromosome number of 24. Now, because again, these were closely related but different species, we do consider this to be allopolyploidy. even though it ended up having 2 new tetraploid species, which is the example that I gave you from the Polyploidy. But again, we define polyploidy either Polyploidy or Aloe Polyploidy based on the process by which it went through, and not just whether more than one species is involved in this, or how tetraploid, sorry, I should say it this way, whether the ploidy number has changed. One thing that I want to talk to you about is that your book does have an excellent animation for this, and you can see the beginnings of it right here. I highly recommend that you go ahead and watch that animation if you're confused at all by what I've talked to you about so far on polyploidy. But to wrap things up on polyploidy, I do want to tell you that, as I kind of mentioned before, when I was talking about the grocery store, that there are many important crops that evolved through polyploidy, including oat, cotton, potatoes, tobacco, and wheat. All of those are polyploid. The example that I'm going to just mention right here is the example of wheat, the wheat that we use for bread. It is an aloe hexaploid. Now, aloe, what does that refer to? Right? You're using the word aloe there, you're saying that it's 2 different species have come together. And you're saying hexaploid, that means it's 6N. So it's not 2N or 4N, it's actually 6N. So, and the way that evolved is very interesting. But what we do know is that today we can produce new polyploids by simply exposing plants to various chemicals to make the errors happen in cell division. And also, just as a refresher for you, that term where you have errors in cell division, that's called non-disjunction. Okay, so I promised you that there were 3 main ways that sympatry could occur. One of them was polyploidy. The other 2 were habitat differentiation and sexual selection. Let's just get a really quick look here at habitat differentiation using an example that I've already talked to you about in this chapter. So sympatric speciation can be the result of a new ecological niche forming. And I've defined ecological niche for you earlier. So the example that I'm going to step you through, I remember I talked to you about the fruit flies, some of which that live on apples. They lay their eggs on apples, some of which lay their eggs on hawthorn fruits. These are called apple maggot flies. And yes, I know that sounds gross, but the baby fruit flies they're maggots, they're just little worms. And sometimes you might encounter one if you bite into an apple. That's perfectly normal. So we're just not used to that because we get most of our fruit from the grocery store, and of course those are sprayed with pesticides. So the North American apple maggot fly can live on either hawthorn trees or on the more recently introduced apple tree. Now, just a little bit of history here for you. Apple trees they're not native to America or North America or South America. They're actually native to Asia and Europe. And so we kind of like a little timeline of when this evolution happened, this sympatric speciation happened, because it happened about the time, or at least sometime after, the introduction of apple trees to North America in the 1700s or so. So we also learned earlier that these flies have habitat isolation. They don't really hang out near each other. So this one is only hanging out near the apple trees, and this one's only hanging out near the hawthorn trees. Because these flies, even though they're not geographically separated by some geographical barrier, they are in fact reproductively isolated with each other by habitat isolation. Interestingly, they are also isolated from each other by temporal barriers, such as, like I said before, with time. So it turns out that the apple maggot fly goes through its life cycle in a time frame where it mates earlier in the year, and the hawthorn flies, they are mating a little bit later in the year, and that's not overlapping. So a really interesting case of these two flies have two different isolating mechanisms that are keeping them apart, even though they don't have any actual geography that's keeping them apart. They've got these isolating mechanisms that are keeping them apart. The last one that I told you we'd talk about for sympatric speciation is sexual selection, how sexual selection can form new species. And sexual selection is how mates are chosen. So the example that I'm going to step you through are the speciation that happened in the cichlid fish. This word is pronounced cichlid, in Lake Victoria, and Lake Victoria is found in Africa. So these are two species of fish, Pundamilia and Nyreii. And if you look at them, you can tell that they are quite different between each other. And of course, in normal light, these two fish can tell the difference between each other as well. Interestingly, Lake Victoria has become quite polluted. Oh, and before I tell you about Lake Victoria pollution, let me back up and say that this is a case of sexual selection driving sympatric speciation because of the appearance of these Pundamilias. Females prefer males that look like this, and the female Nyreii, they prefer males that look like this. So then we have this pollution, and the pollution came about, which increased the opacity, or you just can't see very far inside Lake Victoria anymore. And so it turns out that these two fish look a lot alike in this monochromatic light. And so in the monochromatic light, these two fish do mate with each other, and they do produce fertile offspring. But under normal conditions, they don't. So remember the definition of the biological species concept. These two would not mate with each other. They have a reproductive barrier, in this case a behavioral barrier, and so they have evolved to become more and more and more different from each other. It is also true that due to pollution, these species are actually kind of merging back together and starting to become a single species again. This is not an extremely common event in evolutionary history, but it absolutely does happen. And I'll be talking about that to you a little bit more in the next part. So let's just do a quick review here. With allopatric speciation, geographic isolation absolutely restricts the gene flow of the population. But that doesn't mean that you all of a sudden get new species just because there's a geographic barrier. There also has to be intrinsic barriers to reproduction that have to arise either by natural selection or genetic drift or maybe sexual selection in the isolated populations, such that even if contact is restored, there is no interbreeding between those populations because the reproductive barriers are now in place and they've evolved to be different enough that they won't mate with each other anymore. So that's important. And I usually think about allopatric speciation as geographic isolation happening first, followed by accumulation of mutations that cause these intrinsic barriers to form. With sympatric speciation, the reproductive barrier evolves first, and that reproductive barrier can be through this huge mutation of polyploidy, or it could be due to just sexual selection preferences, could be due to natural selection, it could be due to ecological niches, right, and habitat isolation. whatever the reason, the sympatric speciation, the mutation happens first, and that leads to these other barriers arising. Well, that's all that I have for you for part 3. We'll continue in part 4 in just a minute.